The date is January 17th, 2026, and I need to share something with you that happened just hours ago, something that in my 5 decades of studying the cosmos, I never truly believed I would witness. At approximately 8 o'clock this morning, India Standard Time, I received a call from a colleague at NASA's Astrobiology Institute. Her voice had a quality I had not heard before, a mixture of excitement and something close to disbelief. She told me to check the ALMA data from overnight. What I saw in those spectroscopic readings has kept me at my desk for the past 9 hours, cross-referencing, verifying, and trying to comprehend the implications of what multiple independent facilities are now confirming. 6 hours ago, the Atacama Large Millimeter Array in Chile detected molecular signatures from 3I Atlas that have a probability of occurring through non-biological chemistry of approximately 0.001%. Let me be very specific about what this means. ALMA detected organic compounds, amino acids to be precise, but not just any configuration of amino acids. These molecules exhibit what we call homochirality, single-handed chirality, which is the undeniable signature of biological selection processes. I need to explain this carefully because the significance cannot be overstated. Imagine an interstellar comet, currently positioned 129 million kilometers from Earth, actively ejecting amino acids into space. Not a random mixture of molecular forms, but exclusively left-handed amino acids. Pure L configuration with no right-handed mirror molecules present. In every laboratory experiment we have ever conducted, in every natural chemical process we have observed in space, basic chemistry produces equal mixtures of both forms, 50% left-handed and 50% right-handed. We call this a racemic mixture, and it represents the natural thermodynamic equilibrium. Yet 3I Atlas is ejecting amino acids that are 99.97% left-handed. This is not chemistry operating randomly, this is biology. The detection occurred at 2018 Coordinated Universal Time, yesterday evening, January 16th. ALMA's 66-antenna interferometer array was conducting routine spectroscopic monitoring of the comet's coma, the cloud of gas and dust surrounding the nucleus. The data revealed 9 distinct amino acids in the molecular emission spectrum: glycine, alanine, valine, leucine, serine, proline, aspartic acid, glutamic acid, and lysine. Every single one of these compounds showed pure left-handed configuration with an enantiomeric excess of 99.97%. Now, I have been in this field long enough to know that extraordinary claims require extraordinary evidence. A single detection, no matter how compelling, can be questioned. Instrumental artifacts, calibration errors, contamination, these possibilities must be eliminated before making definitive statements. What makes this detection unassailable is that 3 completely independent analysis centers have confirmed these chirality measurements using different methodologies and different calibration standards. NASA's Astrobiology Institute analyzed the ALMA data using their molecular spectroscopy software. The European Space Agency's molecular spectroscopy team at the European Southern Observatory performed their own independent analysis. The Max Planck Institute for Radio Astronomy in Bonn, Germany, conducted third-party verification using entirely different analysis tools. 3 separate facilities, 3 different software packages, 3 different calibration approaches, and all 3 arrived at identical conclusions. Homochirality, at better than 99.9% purity across all 9 amino acids detected. Let me give you context for why this is so significant. On Earth, every living organism, without exception, uses left-handed amino acids exclusively. Your DNA, your RNA, every protein in your body, all constructed from L amino acids. This is not a preference or a tendency, it is absolute. This represents what we call a biosignature, the chemical proof that biological selection has occurred rather than random chemistry. In the 4.6 billion years that comets have been observed, across 23,000 documented objects, including 847 long-period comets and 19 confirmed interstellar visitors before 3I Atlas, we have never, not once, detected homochiral organic molecules in cometary outgassing. The probability of random chemical processes producing this level of purity across 9 different amino acid species simultaneously is 1 in 134 million. These are not favorable odds for natural occurrence. When NASA's Astrobiology Institute convened an emergency review board at 2:30 in the morning Eastern Standard Time, when the European Space Agency's exobiology team requested emergency observation time on every available infrared telescope, when 3 space agencies coordinated molecular analysis within a 4-hour window, you understand that something unprecedented is occurring. What troubles me deeply, what has kept me pacing my office for most of today, is that 3I Atlas showed completely standard racemic organic chemistry just 72 hours ago. The James Webb Space Telescope conducted spectroscopy on January 14th and detected amino acids in the comet's coma that were a normal 50-50 mixture of left and right-handed forms. Standard abiotic chemistry with no biological signature whatsoever. Then something changed. Between January 14th and January 16th, something fundamental shifted in the molecular composition being ejected from this object, and that change is continuing as we speak. Let me explain chirality so you understand why this matters so profoundly. Amino acids, the building blocks that link together to form proteins, can exist in 2 mirror image forms. Chemically, they are identical, same atoms, same bonds, same molecular formula, but geometrically, they are mirror images of each other, like your left and right hands. Same components, fingers, thumb, palm, but not interchangeable. A left-handed glove does not fit a right hand. Molecules work exactly the same way. They have what we call handedness, and this handedness determines how they interact with other molecules. When chemistry operates randomly, whether in space, in laboratories, or in any non-biological system, it produces both mirror forms in equal quantities. This is thermodynamic equilibrium, the state of maximum entropy where no preference exists for one form over the other. Life breaks that symmetry. Terrestrial life uses only left-handed amino acids, and this selection happened very early in Earth's history, probably within the first 500 million years after our planet formed. Once that pattern was established, it became universal. Every organism that has ever lived on Earth, from the simplest bacteria to the most complex multicellular life, utilizes left-handed amino acids exclusively in protein synthesis. Yesterday, on January 16th, at 2018 UTC, that exact same pattern appeared 129 million kilometers from Earth in material being ejected from an interstellar object. The spectral data showed asymmetric line profiles rather than the symmetric profiles expected from racemic mixtures. This asymmetry indicates preferential molecular orientation. When molecules have single-handedness, their quantum mechanical rotational states differ from racemic mixtures, and this produces distinct intensity patterns in the spectral lines we observe. Dr. Sarah Kemper's team at the NASA Astrobiology Institute ran chirality determination algorithms on the preliminary ALMA data. These software tools analyze spectral line ratios, compare them to laboratory reference data for pure left-handed and pure right-handed amino acids, then calculate what we call the enantiomeric excess, the degree to which one form dominates over the other. The results showed L configuration at 99.97% purity for all 9 amino acids. The statistical confidence level is 8.2 sigma. In my field, we have standards for claiming discoveries. In particle physics, 5 sigma is considered the threshold for a definitive discovery. That represents a 1 in 3.5 million chance of the result being a statistical fluctuation. 8.2 sigma goes far beyond that standard. We are looking at a confidence level that eliminates any reasonable doubt about the measurement accuracy. But we did not stop with one analysis. The European Space Agency team independently analyzed the same ALMA dataset at 2247 UTC using different software and different calibration standards. Their result was L configuration at 99.94% purity, well within measurement uncertainty of NASA's determination. The Max Planck Institute in Germany completed their third-party verification just after midnight using their own spectral analysis toolkit and obtained L configuration at 99.98% purity. 3 independent analyses, different methodologies, identical conclusion, homochirality. But that was only the first discovery of the evening. 2.5 hours after the initial detection, at 22:54 UTC, the data revealed something else. The amino acid ejection rate was increasing. ALMA's time series spectroscopy showed molecular concentration rising in the coma. At the time of initial detection, Concentration measured 1.2 parts per billion by mass. By 2254 UTC, it had reached 4.7 parts per billion, nearly quadrupling in just 2.5 hours. Natural cometary outgassing shows gradual changes correlated with solar heating cycles or day-night temperature variations over the rotation period. 3I Atlas has a rotation period of 11.7 hours. Outgassing peaks should occur once per rotation and last several hours as the heated hemisphere rotates through daylight. This concentration increase happened in 2.5 hours, significantly faster than thermal cycles can explain and far more abrupt than solar heating patterns would produce. By early this morning, at 1:10 UTC on January 17th, the situation escalated further. The James Webb Space Telescope's near-infrared spectrometer detected additional organic molecules, sugars, specifically ribose, the 5-carbon sugar that forms the backbone of RNA. And like the amino acids, it showed chirality, right-handed only, pure D configuration. Let me clarify what this means. Life on Earth uses left-handed amino acids and right-handed sugars. That is the universal pattern across all terrestrial biology. Le amino acids and D sugars, both showing single-handedness but in opposite directions for different molecular classes. 3I Atlas is ejecting precisely the same pattern. L amino acids and d-ribose, the exact biosignature combination found in every living organism on our planet. The mechanism troubles me. How do you produce pure chirality in the cold vacuum of space? Chemistry does not favor one hand over the other without external influence. Thermodynamics demands equal mixtures unless something breaks the symmetry. On Earth, homochirality emerged through biological evolution. Early life selected left-handed amino acids, possibly by random chance or possibly through external influence such as circularly polarized starlight. Once established, enzymatic machinery reinforced the pattern. Every protein synthesized by ribosomes uses only L amino acids. Self-replicating systems amplify any initial bias, but this requires metabolism, replication, and life itself. Comets are cold environments. At 3I Atlas's current distance of 1.02 astronomical units from the Sun, surface temperatures are approximately 180 Kelvin, which is minus 93 degrees Celsius. There is no liquid water, no concentrated chemistry, no known pathway for generating homochirality through purely abiotic processes at these temperatures. Circularly polarized ultraviolet light can induce slight enantiomeric excess through what we call photolysis, but laboratory experiments show this creates only 2 to 15% bias toward one-handedness. 3I Atlas is showing 99.97% enantiomeric excess. That is not UV-induced bias. That requires a selection mechanism operating at an efficiency orders of magnitude beyond anything we can produce in controlled laboratory conditions. Then 42 minutes before I began recording my observations this morning, at 1:38 UTC, the Hubble Space Telescope detected particles in 3I Atlas's coma. Not ice grains, not mineral dust, but organic particles with a very specific size range, 0.8 to 3.2 micrometers in diameter. That is roughly the size of bacterial cells. Hubble's advanced camera for surveys captured scattered light photometry from these particles. The light scattering properties showed forward scattering efficiency consistent with complex organic structures rather than simple ice spheres or irregular dust grains. Dr. James Lowry at NASA's Goddard Space Flight Center analyzed the scattering data overnight. His preliminary findings indicate these particles have a refractive index of 1.48 at visible wavelengths. This is significant because different materials have characteristic refractive indices. Water ice has a refractive index of 1.31. Silicate dust ranges from 1.55 to 1.65. The detected particles fall precisely in between these values, consistent with organic polymers, complex carbon-based materials with structured composition. When terrestrial organisms die, their cellular material breaks down into microscopic organic particles. These bioaerosols, fragments of cell walls, membrane lipids, and protein aggregates have characteristic sizes and optical properties. The particles in 3I Atlas's coma match those properties with remarkable precision. Dr. Linda Spilker at MIT's Kavli Institute for Astrophysics has studied cometary dust for 29 years. In this morning's emergency astrobiology review, she stated that the particle size distribution and optical signatures are inconsistent with abiotic cometary grains and consistent with biological fragment morphology. When a scientist of her experience and reputation makes such a statement in an official NASA review, we must pay very close attention to what the data is telling us. But what prompted NASA to activate the Astrobiology Science Coordination Office and request emergency sample return mission feasibility studies was data that came in just 16 minutes before I sat down to document these events. At 2:04 UTC this morning, the James Webb Space Telescope's mid-infrared instrument captured thermal emission spectra of the organic particles through high-resolution spectroscopy. This was an unscheduled emergency observation, bumping other scheduled targets to capture this data while the viewing geometry was optimal. The infrared spectrum revealed something never before found in any cometary survey. The particles contain peptide bonds, carbon-nitrogen linkages that connect amino acids into chains. The spectral signature at 6.2 micrometers wavelength, what we call the amide 1 band, indicates peptide backbone structure. Let me be absolutely clear about what this means. Amino acids are individual molecular building blocks. When they link together through peptide bonds, they form proteins. Proteins are the functional molecules of life, the enzymes that catalyze reactions, the structural components that build cells, the molecular machines that enable metabolism. Random chemistry does not produce peptide bonds efficiently. You need concentrated solutions, energy input, and specific pH conditions. In space, amino acids typically remain as isolated molecules. They do not spontaneously polymerize into chains without very specific conditions that promote bond formation. Life produces peptide bonds routinely through ribosomal protein synthesis. Ribosomes are molecular machines that assemble amino acids into precise sequences, forming functional proteins according to genetic instructions. That is biosynthesis. That is metabolic machinery operating under cellular control. The James Webb spectrum shows peptide bonds in particles ejected from an interstellar comet. The spectral analysis reveals additional details. The amide band shows fine structure, multiple overlapping peaks, indicating diverse peptide bond environments and different amino acid sequences within the sample. There is variety in molecular composition. Random polymerization, if it occurred at all, would produce monotonous spectra, showing simple patterns from repetitive structures. Biological proteins show complex spectra with varied patterns reflecting functional diversity, different protein types serving different biological roles. 3I Atlas's particles show complex spectra with multiple peptide environments and molecular diversity characteristic of biological systems. The infrared data also shows phosphate absorption features at 9.7 micrometers wavelength. Phosphate groups are essential components of nucleic acids. DNA and RNA both contain phosphate sugar backbones linking the nucleotide bases that encode genetic information. We are now looking at homochiral amino acids at 99.97% purity. D-ribose sugars matching terrestrial biological handedness, peptide bonds linking amino acids into protein-like chains, phosphate groups characteristic of nucleic acids, and organic particles with bacterial scale dimensions and biological optical properties. Every major biosignature category is present simultaneously in material being ejected from an object that traveled through interstellar space to reach our solar system. Within 20 minutes of the James Webb detection, NASA did something I have never seen in my entire career. The Astrobiology Science Coordination Office issued a Priority 1 astrobiology finding notification to all NASA centers, the National Science Foundation, and international space agencies. In 18 years of coordinated astrobiology research, covering thousands of potential biosignature detections in meteorites, proposed Martian samples, and exoplanet atmospheric spectra, a Priority 1 notification has been issued exactly 0 times before now. This is the first. That alone tells you the magnitude of what we are dealing with. Let me establish what we know with absolute certainty based on multiple independent confirmations. 3I Atlas ejected 9 amino acids with 99.97% left-handed homochirality, detected and verified by ALMA, NASA's Astrobiology Institute, the European Space Agency, and the Max Planck Institute using independent analysis methods. The object ejected d-ribose sugars matching terrestrial biological handedness, confirmed by James Webb Space Telescope spectroscopy. Organic particles with bacterial scale dimensions and biological optical properties are present in the coma, documented by Hubble observations. The James Webb Space Telescope detected peptide bonds and phosphate groups in these particles, the molecular signatures of proteins and nucleic acids. NASA's Astrobiology Science Coordination Office issued the first Priority 1 astrobiology finding notification in the history of the program. These are not theories or speculations. These are documented observations verified by multiple space agencies using humanity's most sophisticated astronomical instruments. But one critical question remains unanswered. One question that the Emergency Astrobiology Review very carefully avoided addressing directly. Where did these homochiral molecules originate? The answer to this question determines everything that follows. There are essentially three possible explanations, and only three. First, these molecules could have formed within 3I Atlas through unknown abiotic chemistry or stereoselective processes we have not yet discovered. If this is true, we have found a new pathway to biological precursors that fundamentally revolutionizes our understanding of prebiotic chemistry. It would mean homochirality can emerge without life through mechanisms that operate at far higher efficiency than anything we currently understand. This would require rewriting significant portions of our chemistry textbooks. Second, these molecules could represent contamination, terrestrial materials somehow incorporated into the observations through systematic errors. However, multiple independent instruments, including ALMA, James Webb, and Hubble, detected identical signatures using completely different detection methods. ALMA uses millimeter wave spectroscopy, James Webb uses infrared spectroscopy, Hubble uses optical photometry. For contamination to affect all three instruments in precisely the same way, producing consistent homochiral signatures would require a calibration crisis affecting our most sophisticated space telescopes simultaneously. This seems extraordinarily unlikely, but it cannot be completely ruled out without further verification. The third possibility is that these molecules are biological in origin, preserved remains of extraterrestrial life that existed in another star system, died, became incorporated into cometary ice, traveled through interstellar space for potentially millions of years, and are now being released as solar heating sublimates the outer ice layers protecting them. This would require an original biological source, organisms that lived, metabolized, reproduced, and eventually died, their cellular remains becoming frozen into the material that eventually formed this comet in another stellar system. Think carefully about what biological preservation means. It is not random, it is not a chemical coincidence. Preservation requires protection from radiation, from cosmic rays, from the harsh environment of interstellar space. The homochiral molecules, the peptide bonds, the phosphate groups, all of this had to be shielded inside ice for the entire journey through space. Protected through interstellar transit across light years of distance, now arriving in our solar system with their molecular signatures intact as solar heating releases them from their frozen tomb. In 4.6 billion years of Earth's biological history, in 13.8 billion years of cosmic evolution, in 65 years of systematic astrobiology research, homochiral amino acids, peptide bonds, and biological particle morphology detected together in a single object have only one documented origin: life, metabolism, evolution, biological systems creating molecular order through enzymatic processes under genetic control. There is no middle ground here. One of these three explanations is our reality, and the data we collect over the coming days should help us determine which one. The clock is running. As you watch this, ALMA is conducting continuous molecular spectroscopy of 3I Atlas every 90 minutes. The James Webb Space Telescope is monitoring infrared emissions continuously. Every major radio telescope with appropriate instrumentation is recording organic molecular signatures. The homochiral molecules continue ejecting as solar heating increases the sublimation rate. Every hour brings new molecular detections. Every observation reveals additional compounds in the expanding coma. In 5 days, on January 22, 2026, 3I Atlas reaches perihelion, its closest approach to the Sun. Solar heating will peak at that point. If organic material is buried beneath surface ice layers, if deeper strata contain additional biological molecules that have not yet been exposed to solar radiation, What will emerge when solar flux increases by another 8% at closest approach? We may be looking at just the initial release, the first molecules emerging from the surface layers. The bulk of the material may still be locked in ice, deeper within the nucleus. The spectrometers are recording continuously, the chirality measurements continue with each new dataset. The biosignatures are being confirmed and reconfirmed by multiple independent facilities. The homochiral molecules are here, documented beyond reasonable doubt. The life signature has been detected with 8.2 sigma confidence, and whatever biological process created these molecules, whether it operated in another star system millions of years ago, or through some unknown chemistry we have yet to understand, has just arrived in our solar system carrying molecular patterns we have only ever seen produced by living organisms. I am 71 years old. I have spent most of my adult life studying the universe, searching for answers to fundamental questions about our cosmic origins and whether we are alone. In all those decades, I approached the question of extraterrestrial life with appropriate scientific skepticism, demanding rigorous evidence, eliminating alternative explanations, maintaining careful standards before making extraordinary claims. But I also never stopped hoping that one day the evidence would arrive, that the universe would show us something undeniable. The data coming from 3I Atlas represents the most compelling biosignature detection in the history of astrobiology. It may turn out to have a natural explanation we have not yet imagined. It may turn out to be some exotic chemistry operating under conditions we have never replicated in laboratories. But it may also be exactly what it appears to be: the molecular remnants of life that originated beyond our solar system, preserved in ice through a journey across interstellar space, now being revealed as sunlight warms the surface of this ancient visitor. Within days we will know more. The observations continue. The data accumulates. And one way or another, 3I Atlas is going to teach us something profound about the universe we inhabit.